So this is an example spread, or this is a PowerPoint that I gave one of my collaborators. So if you see something, profound, don't tell anybody. Although I am recording this, so. Oh well. I know the person who I did this for. She's pretty nice, so I don't think. Especially this, I can show you a picture of, So this is the first thing I always do when I give them, when I want to explain somebody's data to them, is I give them a 3D representation of their data set. And remember, we did this for the data set that we did, right? Again, you want to be clear, I kind of outline their groups. You make it as simple as possible. Here are your two main groups. Here's subsets in the two main groups. You can see they're occupying a specific area of space. Therefore, you're going to see differences, right? So usually that's the first thing I do is give them the PCA plot. And I always tell them, this would be a good thing to put in your paper. This is the first thing you put in your paper. Next, I might just give them remember we calculated sources of variation for a lot of the studies we did, or the one we did. That's another thing. I'll give them this picture and say, hey, these are all the factors in your study. These are the things that are affecting gene expression the most. Okay? And that gives them an idea too, especially if they have a factor they're really interested in and it's below this error. Then they can't look at me like, why can't you find anything different? I'm like, because there's nothing different. And you can show them. There's visual proof. So usually I'll do that just to show that, hey, these factors that we applied to the data set truly did make a difference on the gene expression. And you kind of see that with the PCA as well. But this is more of a concrete way to do that. And then everybody loves PowerPoints. And I'm sorry this isn't showing up as better, heat maps. So again, that's what I'll do is I'll show a heat map. I'll take, say, the top 30 most significant transcripts, make this heat map, this color scale to show them what the gene expression is doing in each of their different groups. And again, I made this with Partex, so you all can do this as well. You want to get specific, you can show them individual gene expression. Yeah, I might have to cut this slide out. These are actually, they're really interested in these genes. So this study, they're looking at ischemia, and, if they give them this drug, these animals that they give ischemia to, the stroke, they seem to recover better. And they're wondering what's going on, when they manipulate this gene. Here are some of the genes that, change. This SOD is superoxide dismutase, which actually fights, is a scavenger of free radicals in your system, right? This changes, this is important to it. And again, dot plots, this is a dot plot, which is, a really good way to show this. We can make the different groups, different sizes, different shapes. You can see the box plot here, and I just want to explain this as well, that the line here is the mean of the gene expression for that group. The middle box is the middle 50%, and everything else on the side, I believe, is the quartiles, maybe. I think, what is it? 95 and, I think it's 95 and 5%. Or maybe not, I can't remember. No, that's right. Yeah. Okay. And then you can include Venn diagrams. How'd you get the gene list? What genes are you looking at? What are some of the similar genes between our different variables in our study? I love Venn diagrams. Venn diagrams are cool. You can do the comparison analysis as we kind of go over. You include these into your presentation. Here are your different gene lists. Here's how they compare. You can tell these two are very similar, and this one is very, very different. And in fact, it's the opposite, and we kind of like that if you knew the study. But again, this gives them a very easy way. Remember, again, orange and blue. If it's orange, that means it's more active. If it's blue and inhibited. A very easy way for them to just kind of look at these pathways and go, what's going on in my system? Oh, NOS signaling is up in these guys, but not in this one, And then after that, once you've kind of explained everything, you've given them an idea of some of the functions, the diseases that are overrepresented in the gene list, then you go into your specialty stuff, right? This is kind of a lot of people will just stop there. They'll just go, whoa, that's fine, Mike. This is a grant study, or this was just kind of part of the other study. It's not that big of a deal. We don't want to get too far in the weeds on this, or we don't want to give up too much because, we're still working on this. We don't want to show them what we got. But I always, dig in farther to the thing, and then you can pull out, just some, kind of regulatory networks from here, and these are just examples of some of the things I saw in this data set, and they didn't use this one. I think they did use this one. But again, and these are some of these pathways, we've made our pathways ourselves. This is some of using the grow function and connect to make some of these that are specific to questions they had. And that's kind of what you do, is you want to give them all the information. You want to take all that great information from IPA, take that, put that into an Excel spreadsheet somehow, right? You can do it in many different ways, but you want all that information there so they can sort and they can search it. Then you also want to give them diagrams and graphs on some of these pathways and networks that we come up with. All right, any questions?